Hello, I'm Jesus de la Fuente. I'm a first year PhD student under the supervision of Dr. Miquel Hernández and Dr. Idoia Ochoa in Tecno San Sebastián, Spain. And today we're going to be presenting the identification of transcriptional network disruptions in drug-resistant prostate cancer with TraRe. So this all started with a promoted clinical trial from Mayo Clinic we are working on. It's a metastatic castration-resistant prostate cancer project in which we have a bulk RNA dataset containing 21K genes and 46 samples. So these 46 patients has been sequenced before they were given abiraterone, and the time to treatment change has been used in order to label them as responders or non-responders to this abiraterone. So our motivating research question was, can we detect regulatory network differences, what we call rewirings, between different phenotypes, in our case, between responders and non-responders patients? So we knew that the traditional approach was to look for differential expressed targets that have a strong global correlation with a transcription factor in order to establish a relation between this target and a regulator within the network. So here we have this target gene that is differentially expressed among this binary phenotype, and it's also globally correlated to this regulator. But I wanted to focus on different cases in which network differences can be can appear within the network. So here, for example, we have this target gene that it's not differentially expressed nor globally correlated to this regulator, but in fact, if we take a look at the local correlation, the correlation among phenotypes, in fact, we can see a disruption on the relationship. So in order to go farther into the study of these cases, we're going to be working with gene regulatory networks that we believe that they provide a concise representation of the transcriptional regulatory landscape of the cell, even though that the reverse engineering problem that we are facing of inferring gene regulatory networks from gene expression data is still a major challenge because of the complexity to infer the regulatory programs of the underlying biological processes. So we're going to be working with a model-based approach for building GRNs. So model-based approaches, we believe that they are one step ahead from the single gene networks. So the single gene approaches, if we model a regulatory network as a graph in which nodes are targets and regulators, single gene network approaches connect targets and regulator if they have an individual relationship. But model-based approaches consider that within the network, we can have multiple drivers, multiple transcription factors in which a combination of them may be regulating, maybe explaining the expression of a target gene. So we consider this model-based approach more realistic for individual pathways, processes, and also more useful from a system biological point of view. So what are we looking for? This is what we call a Rewired-GRN. So figure on the left shows a built gene regulatory network from all these samples, the 40 samples of a bulk RNA dataset. And when we unravel particular samples from a phenotype and we build the gene regulatory network associated to these ones, we can see in fact here that edges between drivers and target are getting switched off from one phenotype to another one. So this yields disruption in the networks, disruption in the gene regulatory networks. So this is what are we looking for? What are we expecting to obtain within the inferred gene regulatory networks? So how to infer these GRNs? How are we doing this? We have developed TraRe, that it's a computational method to infer transcriptional rewiring of GRNs associated with a phenotype. So TraRe consists on 4 blocks. The first one, it's the model-based gene regulatory network inference tool. So it takes a bulk RNA dataset and it infers a number of gene regulatory networks. So from the inferred gene regulatory network in the form of modules, these modules are going to be fed into the rewiring step, in which each module is going to be evaluated and it's going to be labeled as rewired or not. Also, similar rewired modules are going to be grouped into what we call robust rewired GRNs. And this aggregation of rewired GRNs are going to be visualized in order to check for the disruption we are looking for. Also, in order to evaluate whether specific edges are getting disrupted, we're going to output also a summary in the form of a table where we can take a look at a specific disrupted edges. So from the bulk RNA dataset, the first step, the module generation is going to work on 2 phases. So phase 1, it's going to work similarly to k-means, and it's going to iteratively place targets and drivers that are similarly co-expressed into modules. So note that these modules do not have connections between drivers and target genes. So this is what is going to happen in phase 2. By using a variational bias spike regression, local contribution of drivers to target genes are going to be drawn in the form of edges. And these edges are going to represent the local contribution these drivers are having with respect to these target genes. So for each target gene, this model is going to be fitted and it's going to try to explain the expression of the target gene as a combination of the drivers within each module we have previously inferred. So once we have all the modules, all the gene regulatory networks that have been inferred in the form of bipartite graphs, these are going to be evaluated in the rewiring step. So the rewiring, it's a permutation test which have as a null hypothesis that the covariance matrices are equal. So from samples of phenotypes, covariance matrices are going to be computed and a dissimilarity metric is going to be computed as well. So this dissimilarity metric, it's informative about, it's based on the Frobenius norm of the differences of the covariance matrices. This module is going to be rewired if this metric, when evaluated on the chosen phenotype, is significantly larger than a number of fixed permutations, a number of samples that are going to be random labeled, are going to be evaluated against the chosen phenotype. So in order to increase the generalization of the results, we're going to be randomly sampling 80% of patients on each run. We're going to be running TraRe multiple times to discover robust GRNs. So here, for example, we're having rewired modules, and similar rewired modules across runs are going to be grouped together when applying a hierarchical clustering method. So originally, a consistent rewired GRN will be yielding two similar rewired GRNs across runs. So this similarity, it's going to be representing on this adjacency matrix. And from here, this behavior is going to yield robust rewired GRNs. Notice that also we can build a graph from this adjacency matrix we can apply graph clustering techniques from these modules and obtain also robust rewire-GRNs. So if we run TraRe 10 times, if we perform 10 runs over the promote bulk RNA dataset, we're going to obtain some modules. And if we perform again the rewiring, some modules will be labeled as rewire. If we perform a graph clustering technique over the infer modules, we're going to obtain communities. In our case, we obtain 93 communities, where 11 of them contain one or more rewire-GRNs. So we chose 9 of them, and we functionally annotated by using gene set enrichment analysis. So here we wanted to focus now on communities that only contain rewire-GRNs. And in order to do this, we increased the number of rounds up to 50, and we built a graph from all the rewire-GRNs. So each node here is a rewire-GRN, and an edge between 2 nodes indicates that they are they contain high overlap. So they're really similar to each other. So if we take a look at the dark green community, here we're having that this community belongs to a very consistent module that it has been labeled as rewire across all runs. Each of these modules are very similar to each other. It has been selected to be the same community. So if we focus on this consistent, robust rewire-GRN, we saw that the regulatory structure was lost in non-responders. And here we asked ourselves another question. So are these transcription factors specific to rewire modules? Are we expecting to obtain the same transcription factors if we perform the same experiment on non-rewire modules? So in order to do this, we built a graph from each rewire driver, of the rewire modules. So this table is informative about whether a driver is or is not on a rewired or non-rewired module. So from here, we perform an hypergeometric test and we obtain a p-value. So we use this p-value to rank the drivers from the rewired modules we obtain. And in a similar way, we could also evaluate Regulons, which are transcription factors and its associated target genes. So we could evaluate these Regulons to be repeated within rewired modules. So now we try to validate if disruption was detected on drivers from the previous list we have generated. So from this list, after knockdown of the top TFs from the list, expression level of 4 genes were found to be significantly changed by the quantitative real-time PCR on 2 pairs of parental and average-resistant prostate cell lines. These target genes are related to the cytokine chemokine pathways. So this has been the identification of transcriptional network disruption in drug-resistant prostate cancer with TraRe. I hope you have enjoyed the talk, and I hope it's all clear. I will be really glad to be answering any question you have. Thank you very much. This is all from my side. Have a good day.